As I have been talking again and again, then whenever we are talking about any pathway, any chemical reaction which is taking place in body, one of the most important aspect we have always to keep in mind is that how that process is regulated. We don't need all the time focusing on what is the end product, what is the intermediate product, what is the reactant, what is the rate of reaction, so on and so forth. These of course are important things, but even perhaps more important is that how the reaction as a whole is being regulated by the different machineries of the cell, different machineries, molecular machineries in our body. So, it is very important. So, gluconeogenesis is not an exception, it is a very complicated process, and it also has to be regulated, because I have already discussed with you that how important is the presence of glucose for your body. Regulation of gluconeogenesis is determined by how much it is very much controlled by hormones also, like glucagon, and in addition to the glucagon, how much the substrates are available which are involved in the formation of different products of gluconeogenesis. Then the certain adaptive changes which takes place in the enzyme also, in the activity of the enzyme also, that is also important. Of course, I have already discussed that, and we will discuss in detail just shortly that how much the hormonal control is important for the regulation of the process of gluconeogenesis. One important aspect is that how glucagon affect different effector molecules which takes place, which are part of the process of gluconeogenesis. One of them is that it lowers the level of fructose 2,6 biphosphate, and this decrease in the level of this compound leads to the activation of fructose 1,6 biphosphatase, and it also inhibits phosphofructose kinase 1. All of these things leads to the more and more formation of glucose through the process of gluconeogenesis. The covalent modification of the enzymes which are involved in the process of gluconeogenesis, it is also very important way of modulating the regulation of a process like gluconeogenesis. For instance, here glucagon as a part of the pathway, it binds with the G protein coupled receptors, very major receptors which are involved in the cellular signaling. They are present in the cell membrane, you all know about them. This interaction between the receptor and the hormone then leads to the conversion of protein hepatic. Keep in mind, we are talking because all the time whenever you are talking about the process of gluconeogenesis, it is very much taking place in liver, or very much the enzymes which are part of which are responsible for the process of gluconeogenesis, they are present in the liver. So, that is why we are using the word hepatic protein kinase. So, the hepatic protein kinase is inactivated because of this signaling pathway, the signaling pathway in which glucagon interacts with the G protein coupled receptor, which in turn as it's a classical pathway of G protein coupled receptor signaling, in which G protein coupled receptor gets activated. It is activated cyclic AMP then affect protein kinase, and the inactivation of protein kinase then leads to the decreased conversion of pyruvate into molecules which leads to the process of, let's say, ATP formation like Krebs cycle or other, it actually leads to the reversal of the process of glycolysis, that is the process of gluconeogenesis is enhanced because of these signaling pathways. So, in this way, for instance, the inactivation of protein kinase here leads to the diversion of phosphoenol pyruvate towards the formation of glucose synthesis. Another way of regulating a pathway is that certain enzymes or certain hormones effect very downstream of the cellular signaling. What is the most downstream effectors of the cellular signaling? These are the genes. So, the modulation of the genes, modulation of the transcription factors, which leads to the regulation of the transcription, it is also one of the strategy through which the enzymes or hormones control a pathway. So, here again the glucagon increases transcription of phosphoenol pyruvate carboxykinase gene, and as you have already learned, this PEP carboxykinase gene leads to the formation of phosphoenol pyruvate from pyruvate, which enhances the process of gluconeogenesis. Insulin, on the other hand, acts in reverse. It is, we can say, antagonistic of the processes which are activated by the glucagon. So, more and more insulin, lesser will be the gluconeogenesis. More and more glucagon, more will be the gluconeogenesis. Then finally, there are certain substrate molecules also which are precursors molecules which are involved in the process of gluconeogenesis, which you require because there have to be some substrates which will then be used in the formation of glucose in the process of gluconeogenesis. Some of the most important of these are the glucogenic amino acids which influence the rate of glucose synthesis in the process of gluconeogenesis. Insulin, जैसा कि मैंने पहले कहा, will affect it negatively. So, more glucagon you have and more you will get the substrate for the process of gluconeogenesis. Insulin, on the other hand, inhibit this process. ATP and NADH are also certain compounds which are act as the substrate for this process. And then as acetyl-CoA, keep in mind that when your body is under starvation, what will happen that because you have lack of glucose, then there will be increased level of lipolysis, and then you have more and more fatty acids present in your liver, and as a result these fatty acids now will be stimulating the formation of glucose also. And in this way we can say that more fatty acid formation leads to more glucose formation because of the enhancement of what we say the process of gluconeogenesis. What is the mechanism behind this? Is when more fatty acids are produced, they give rise to more and more acetyl-CoA, which is the unit of the fatty acids, and acetyl-CoA then inhibits the PDH, pyruvate dehydrogenase complex. When it inhibits the pyruvate dehydrogenase complex, it definitely then diverts the pathway towards the formation of phosphoenol pyruvate, because pyruvate cannot be converted into the products which are used in the Krebs cycle. So, definitely then the process of glycolysis will be reverted back, and in this way you will get more and more glucose.